So now we're going to talk about a new phylum. This is the biggest phylum that we've talked about so far. Um, Prior to this, most of the phyla, most of the organisms you probably never heard of or seen before. Now we're going to start getting into organisms that you might be more familiar with. So let's talk about the mollusca. And so again, if we look at our phylogenetic tree, there's lots of phyla that we're skipping past. And it's not because they're uninteresting, and it's not because they're unimportant. It's just that, we don't have a lot of these specimens, and we don't have a lot of time. We did talk about the Ectoprocta. We did talk about the bryozoans in our last video, in our last lecture. And so, look at some of these in your book. There are some very interesting organisms here, but we're going to jump ahead to the mollusca. And we've already talked about some mollusks before. Um, Again, you're probably familiar with some of these. You've seen these before. You remember from last week, we talked about the trematodes or the flukes, and we said they often have several different stages in their life cycle, and often some of those stages require a mollusk, right? And so here's an example of a human liver fluke, and it requires a gastropod, a snail, for several of the forms within its life cycle. And we saw this with a lot of the flukes. So we've already talked about these in a little bit. We'll talk about them in more detail. Like I said, this is a huge phylum. You've got lots of species. They're very successful. You also got lots of them in the fossil record that have gone extinct. So they've been around for a long time. Um, they're important to the history of life on Earth. Now we see a lot of these in the fossil record. Why might that be? What's one reason that you can think of that you'd see a lot of things like snails in the fossil record? Well, it's because they have hard parts, right? And so hard parts tend to fossilize better than soft parts. And so there are certain types of organisms that you just have a better fossil record of because they fossilize better. And the mollusks are certainly one of those particular groups of organisms. Now, you might ask, well, do they seem to be successful just because they're in the fossil record because of these hard parts, and maybe they're not always been successful? That's probably got something to do with it. But also, if you just look at modern day organisms, you see that a lot, the mollusca are very successful. So it's a combination of both. But we have a good fossil record of them because of these hard parts. So here's a figure from your book kind of showing the different classes and the relative numbers in each class over time. And so if you look along the bottom, gives you time in millions of years ago. So you've got here 570 million years ago, 245 million years ago. And so you can see the ancestral mollusk was, maybe 600 million years ago or so. And then the width of each branch here is relative to how many genera or families of that organism, of that class, were around at different times. And I like graphs like this because they really help to kind of give you a picture of what the world was like at different points in time. And so you can see something like the squids here, cephalopoda, and 300 million years ago, there were a lot more of them than there are today. And there were a lot more of them relative to other mollusks. You can see the bivalves began to differentiate a little bit more once the cephalopods became less numerous, and then they've stayed fairly steady up until the present day. And you can see the gastropods have steadily increased over time, and you get more and more of them. So this just gives you an idea of the relative numbers of each of these different classes. Now, we're going to concentrate on these three classes because they're the biggest and the ones you're most likely to run into here in Kentucky. Again, these other classes are interesting and important, but we just don't have time. You do need to know the names of these three classes just because they are so common. And that's the gastropods, the cephalopods, and the bivalves. And we're going to talk about each of those in a little bit more detail right now. Here's a generalized gastropod body plan, but it's got a lot of the things that are common to all mollusks. And we want to contrast this with the organisms we've already looked at, and you can see that that we're getting more differentiation. We're getting more complicated organ systems and things like that. Note that now we have a coelom, right? Last week the flatworms were acoelomate. They formed the mesoderm, but they didn't form that cavity in the mesoderm called a coelom. Now we have a coelomic cavity, and most of the organisms from here on out are going to have the coelomic cavity. So this is something that developed and was a huge advantage to a lot of these organisms. Well, what's the advantage to having that? There could be many. For the mollusks, it just allows for packaging of the organs, and the organs are better protected, and it allows them to work more efficiently. So in some other organisms, the coelom is important for like a hydrostatic skeleton. So having this coelom seems to be an advantage, and that's why it's spread in several of the other phyla. Note that in the mollusks, or in this gastropod we're looking at, you just got the gonads and the heart in the coelom, but some of the other organs like the stomach are not in the coelomic cavity. But then if you think about your body plan, you know, most of your organs are in your coelomic cavity. So you can see that this is sort of an early version of the coelom, and then you can also kind of imagine how this evolved into later organisms. The mantle is a structure that's unique to the mollusks. It's just these folds of skin, and the mantle secretes the shell. So again, if you think back to things like corals or bryozoans that secreted kind of an exoskeleton, this is the same kind of idea, but now it's in a later on organism. And so the mantle is what secretes the shell, and it also creates this mantle cavity. And so the mantle cavity is kind of a unique cavity to the mollusks, and it's similar to a coelomic cavity, but it usually houses the gills, is the only thing you're going to find in the mantle cavity. Another structure that's unique to the mollusks is this radula. And most of them have it, and it's up here in the mouth. And it's sort of like a tongue with teeth. It's a specialized tongue that can be used to scrape algae and things up. And these teeth, break off, and then our new ones move in and replace, and they're constantly producing new teeth on the radula. So this is another structure that you need to know that's unique to the mollusks. Now we have gills, which we haven't really seen in any organisms, but in the mollusks and in other organisms that we're going to talk about later, we'll start to see more gills, which of course are important for gas exchange in aquatic environments. Since you're trying to get gases to diffuse into the blood from the water, and you're getting other gases to diffuse out of the blood into the water, the gills need to be very thin. And so you've got very thin, like one cell wall thick tissues, which makes them very delicate, which makes them very easy to get damaged. And so this is a sort of a soft spot, sort of an Achilles heel for a lot of aquatic organisms. And so that's why you see things like the mollusks tuck these inside into the mantle cavity to try to protect them a little bit. And when we look at gills in other organisms, we're going to see similar characteristics, that they're very thin and delicate, and a soft spot in a lot of organisms. Notice in this figure from your book, they're showing something called a countercurrent mechanism. And we see this in a lot of aquatic species, and we see this in a lot of species that have gills, and we see this in a lot of different areas, not just with gas exchange. But basically what they're trying to show you is that the blood is pumped in an opposite direction as the flow of water. And so the organism, the mollusk here, is constantly pumping water across these gills, right? Because you've got to bring in fresh oxygen, and then you've got to get rid of that water that you put carbon dioxide into. No different than you breathing in and out. And so as you pump that water across the gills, it flows in one direction, but usually the blood inside the gills flows in the opposite direction. And that's called a countercurrent mechanism. And we see that for things like gas uptake. We see that for things like heat retention in the body. You see this in a lot of animals. And we're going to talk about it in more detail when we get into like the vertebrates, and we talk about fish specifically is another place where we see a lot of these countercurrent mechanisms. But it basically increases the efficiency of gas exchange, or it increases the efficiency of temperature retention, or whatever you're using the countercurrent for. So in this example, what that means is the mollusk can get more oxygen out of the water simply by having this countercurrent mechanism. And so there's more oxygen available, and that's a big advantage. So we'll talk about countercurrents several times in the upcoming weeks. Now you also notice that we have more of a circulatory system where we have a heart now, but it's not as complete as, say, a vertebrate circulatory system. It's an open circulatory system. So if you look at this figure here, you can see that here it is. You can see that the heart pumps, and it takes blood from the gills, and it pumps it, but then it just sort of pumps it out into the body. And then it just kind of sloshes around the body as the snail moves. That's an open circulatory system. And so it's not great at moving oxygen around the body because you don't have a lot of blood pressure, and you're just kind of counting on the blood to sort of slosh around and then make its way back to the gills on its own. And so you can sort of see that this is an early circulatory system that works better than no circulatory system, but it's not as efficient as a closed circulatory system. But it's fine because we see this in something like a gastropod, a snail or a slug, and they don't run very fast, right? Snails are not known for their speed. They don't move very much, and so they don't need as efficient a circulatory system. Or conversely, you could make the argument that because they have this inefficient circulatory system, they never evolved the ability to move faster. Either way, it's sort of the same idea. So again, the gastropoda are snails and slugs, and a huge number of species that you're familiar with. A slug basically looks just like a snail without a shell on it, right? Gastropoda, gastro means stomach, poda means foot, so these are the stomach foots. And this is just the way that it was named earlier. The foot, the big muscular structure that's used for locomotion, which you see down here at the bottom, it's down on what early zoologists considered the belly. And so that's where you get stomach foot or belly foot. That's why it's called gastropoda. Okay, so another class that you're probably familiar with are the bivalves or bivalvia. And so these also have a shell, like the gastropods, whereas the gastropods have a one-piece shell, bivalves have a two-piece shell, hence bivalva, two-piece shell. Now the bivalves don't have this radula that most of the other mollusks have, and they also don't really have a head, right? They don't have cephalization so much. And so you're familiar with these. These are things like clams and oysters and mussels. And if you picture that, you can picture there's not really a head. So it's just in a different class, different kind of mollusks. Now, these are probably some of the most endangered animals on Earth, certainly in the US. These are some of the most endangered. And you can argue about, who's more endangered and who's less endangered. And at some level, it's a pointless argument because an endangered species is one that is very likely to go extinct unless we actively do something to stop its extinction. And this is a group you tend to see a lot of bivalves and freshwater mussels specifically that are terribly endangered. And a big reason is because they're not very mobile and they're filter feeders. And so they're dependent upon good water quality. And so if the water quality is bad, so if there are toxins in the water, or if there's lots of sediment in the water, or if the oxygen gets low in the water, whereas a fish or an insect, a lot of other mobile organisms can then just escape and try to find better habitat, the bivalves are stuck there. They can't move very much. And so consequently, poor water quality has a disproportionate effect on this group of organisms. We also see a lot of loss of aquatic habitat, just straightening channels, diverting water, damming up water. So a lot of our streams where these organisms are found have been severely impacted by humans. And consequently, the bivalves don't do well because they've lost their habitat. Another reason that they're highly endangered is because they often require another species, usually a fish, for their reproduction. And a lot of the fish that they rely upon are also endangered for a lot of the same reasons. And so, if you lose that fish, then you also lose the mussel because the mussel needs the fish to reproduce. And so, when we talk about things like poor water quality and degradation of natural water bodies, these are some of the organisms that are greatly affected by that. A final reason has to do with commercial exploitation. We used to make buttons out of these. And so this had a dramatic impact on the mussel species in things like the Mississippi River. Excuse me. And although commercial exploitation is not as bad as it once was, a lot of that is simply because these have been decimated and there just aren't enough to make it commercially viable. So if we look at a bivalve, you see it's quite different than a gastropod. You've got the foot, again, is sort of toward the bottom here. And whereas the gastropod uses that foot and cilia on that foot to kind of help pull themselves along, the way the mussel moves is it pumps blood into the foot, which causes it to swell out. And then the foot can expand into, cracks in the rocks or between rocks and things. And then there are muscles attached to that foot that allow, the foot acts as an anchor, and then the clam or the mussel can pull itself forward. And so it can very slowly move by extending that foot and then using that foot as an anchor to pull themselves forward. You see you've got a couple of siphons here, an in-current and an ex-current siphon, and this is how the bivalve sucks water in, runs it across the gills. Also, not only are they taking out, oxygen and getting rid of carbon dioxide, but they're filter feeders. So they're also filtering the water as they suck the water in and then they blow the water out. You can see on this picture there are growth rings, and you can use those growth rings to actually age the mussel. So you can count the rings and figure out how old it is. If you look on the inside, this is the inside of the shell, and you see some spots that are very obvious in the shell. And these are places where muscles attach, and the muscles that are used to pull the shell closed. And then you've got this big opening down here. This is where the mantle cavity is where you're going to find those siphons. And this is what we're going to be dissecting in class, so we'll get to see all this in great detail. Here is a kind of a side view of those muscles, and you could imagine you saw in the previous picture where they attached. And so when those muscles contract, you've got a hinge up here at the top of the shell, and it pulls the shell tightly closed. And then when you relax those muscles, that ligament has got some tension in it, and so that ligament can cause the shell to spring open. And so that's how the clam or the mussel can open and close itself. Again, they've got a mantle, which are these folds of skin that sort of surround the viscera, and again, the mantle is what secretes the hard outside shell. But the mantle in the bivalves is also known for the production of pearls. And so if you get some kind of an irritant, like a grain of sand or something, trapped underneath that mantle, the mantle will sort of grow around it and secrete these substances to kind of wall off that irritant so it can't get into the bivalve and cause trouble. I guess it's kind of like in some way, it's sort of like an immune system response, although it's not really the immune system. But this is how a pearl forms, is by that mantle secreting these substances around that irritant. And so to create pearls or to culture pearls, you can purposely put irritants in these clams and they will produce pearls. And it turns out that you can make really good pearls by taking the shells of freshwater bivalves, crushing them up, and then sticking them in Asian oyster species. And for whatever reason, this causes those oysters to make really nice pearls. And so this is a great way to make pearls. Um, Unfortunately, it takes a large amount of freshwater mussels to grind up to make that irritant. Well, where do you get the freshwater mussels from? You get them from a place like Kentucky Lake. And for a while, this had a huge impact on the mussels in Kentucky Lake because there was a huge commercial market for them, and people were taking tons of them out to sell so people can use our mussels to culture pearls. Um, but I think the regulations have changed since then because they had a huge impact and mussel levels were really declined because of this in Kentucky Lake. As I mentioned, the mussels usually need a fish species to complete their reproductive cycle. And so they release a larvae called a veliger, and that's just the gastropods do that too. That's part of the reproductive life cycle. In the bivalves, this veliger turns into a different larval stage called a glochidia. And the glochidia swim up and parasitize fish gills. And so they live in the gills of fish where they can absorb nutrients. You know, it's a blood-rich area, a nutrient-rich area. So they act as a parasite of these fish until they get big enough to drop off. And then when they drop off into the sediment, then they grow into an adult mussel. And so this is an important way that these organisms reproduce. And as I mentioned earlier, often uh, a particular species of mussel requires a particular species of fish. They can't just use any old fish. They need a particular species of fish. And so if the habitat is degraded and that fish is gone, then you're going to lose that mussel as well. So this is a big problem in aquatic ecology. And like I said, mussels and other bivalves are greatly imperiled. Um, this is important because they are indicators of good water quality. Why are the mussels disappearing? Because the water quality is bad. But we all need good water quality. So we use these as indicator species. And so if you have a stream that's devoid of mussels or of certain types of mussels, that suggests that that stream has poor water quality. And we all need clean water. And so this is something that we need to pay attention to. They also need to be in the water just because they're an important part of the food web. They're an important food source for a lot of organisms. There are a lot of fish, like freshwater drum, that like to eat these mollusks. Shellcrackers or red ear is another fish that likes to eat these mollusks. You think about things like raccoons and otters, they love to eat these mollusks. So they're an important part of the food web. And as I mentioned, they're filter feeders. And so they filter the water and help to clean the water, which is important for things like primary productivity. So they're an important part of the food web, and this is why we're very concerned about their losing a lot of these species. I posted a link to this video. I'm just going to show you a couple of still grabs from it, just to show you how much these bivalves can filter the water. And so you've got two tanks with, at the start, have identical water in them, one that has clams and one that doesn't. And you can see that after a couple hours, the one with clams is a lot clearer. This just goes to show you the incredible filtering power of these mussels, which is one of many reasons why they're important. All right. Those were the bivalves. The last class that we want to get into in a little bit of detail are the cephalopods. And so this is squids and octopuses and cuttlefish and the nautilus. And so these are organisms that you're probably somewhat familiar with. We don't really have any of them here in Kentucky. These are all marine species. Now, whereas the gastropods were somewhat mobile, snails can move, but they're not known for their speed. Slugs can move, they're not known for their speed. So they're somewhat mobile, but they're slow and they're herbivores. And the bivalves are not very mobile. Again, they're mobile, but they're probably less mobile than the gastropods. And they're filter feeders. Cephalopods are fast and they are active predators. So they have a very different lifestyle. Now, before we go any further, yes, it is octopuses. A lot of people want to say octopi because it sounds right, as far as Latin goes, but it is not correct. Octopuses is the correct plural of octopus. All right. So here is a generalized cephalopod body plan. This is a squid. Um, and so ceph, you remember from cephalization, ceph means head and poda means foot. So instead of the gastropods, which were the stomach foot, these are the head foot. And so basically the head is down near where the foot is. And that's kind of where they got their name from. The foot is different than the foot in the gastropods and in the bivalves, right? It was a big muscular thing. Here in the cephalopods, the foot is modified, and this is how you get the tentacles. And so instead of having that big muscular foot like in the bivalve, or that foot that runs along the ventral surface that is used for locomotion, in these, that foot has been modified and you get tentacles, and you also get this funnel, which is used in swimming. All right. And so then you also note the location of the radula, which is labeled. It's not labeled on this one. But here's the mouth, and here's the, oh, here it is. Here's the radula, of course, which is in the mouth, and the brain. And so those are found in the head. And so that's kind of the generalized head region. It's not up here. It's not on this end. It's down here. The head is sort of down near where the foot is, and so head, foot, cephalopoda. And so, again, these are active predators that are much faster than the other classes, and they are faster because they have jet propulsion. And so they can use muscles to push water out of this funnel, and that makes them very fast. So you can see how they have a very different body plan, and so consequently they have very different behavior. Now, whereas in the snails and the slugs, in the bivalves, where you don't move very much, you've got an open circulatory system because it's not very efficient, but you don't really need it to be that efficient because you don't move that much. These guys move a lot. They're very fast. They need more efficiency. So they have a closed circulatory system. At least it's more closed than in the other classes. And so this keeps the blood pressure up. It helps to pump the blood around and move the blood around more so you can get oxygen where you need it because you're a much more active predator. The problem is it's all kind of backwards. The blood goes to the body first, and then after it goes to the body, then it goes to the gills, and then it comes back to the heart. Whereas if you think about your circulation, you know, the heart pumps blood to the lungs, it picks up oxygen, it gets rid of carbon dioxide, and then it pumps it to the body. Here, I'm going to back up one. Here, we're doing it a little bit backwards. We're going to the body first, and then we're going to the gills. And consequently, by the time the blood gets to the gills, it doesn't have a lot of blood pressure. And so to compensate for that, they have a second heart. And so you see the, where's the label? Here's the systemic heart, right? And you can see, you follow that it pumps blood to most of the body. Then that blood makes its way back to the gills, and you've got a branchial heart that helps to increase the blood pressure to the gills, and then it makes it back to the systemic heart. So you've got a couple of hearts in here to try to help compensate for that low blood pressure. Why is low blood pressure important in these gills? Well, I mean, think about the gills. They're very thin. They've got lots of capillaries. So they got very small capillaries of blood because you need to do gas exchange. That's why they need to be small. But those small blood vessels, those capillaries, have a lot of resistance. And so it's hard to pump blood through them because they're so small, right? It's like pushing water through a small hose instead of a big pipe. And so you need higher pressure to get the blood to go through there. And so to get that higher pressure, you have a second heart in these cephalopods. And like I said, they're more active than other mollusks. That's why they need this more closed circulatory system. So here you're looking at a nautilus, which is a very interesting looking organism. And here again, you see the radula and the mouth down here near the foot. And the foot has been modified to make tentacles and the funnel. So that's where you get the cephalopod from. You see how the gills are down here in this funnel, which is part of the foot, and then of course the mantle cavity that we've been seeing is where we always find those gills. And so here you find the same structure in this, these are still mollusks, but they're just organized a little differently. Now, whereas the gastropods and the bivalves had a heavy shell, the cephalopods tend to not have as heavy a shell. Now, the nautilus of all the cephalopods probably has the heaviest shell, but you see all these chambers, and all these chambers are gas-filled, and so that gives it buoyancy. So although it has a heavier shell, it's more buoyant than the other mollusks because it's got these gas-filled chambers. Now, if you look at a cuttlefish, they have a less well-developed shell, and it's mostly internal. Then if you go to the squids don't have much of a shell at all, but they do have this pin. They do have a stiff rod that sort of runs along the dorsal surface. And we'll be able to see this when we dissect these squids. And so, again, it's the remnant of a shell, and it gives them some stiffness. But you see in each of these organisms, you're getting less and less of a shell. And then when you get to an octopus, octopuses don't have any kind of a shell. So you can see kind of the subtle differences throughout this class. A lot of the cephalopods have a very strong beak. Here we go. Again, down there at the mouth, and we'll look for this when we dissect them. And so this beak is used like a bird uses its beak. It's used to grab and hold and cut up prey. The cephalopods have the largest brains of any invertebrate. And so they're well studied because they have these large brains. They also have very large nerve cells. And some of the first work done on understanding how nerve cells work was done using cephalopods because they have such well-developed neurons and such big brains for an invertebrate. So again, compare this with some of the other phyla. We've been looking at phyla that have more simple, like a nerve net, and then you see a little bit more complexity in the flatworms, and now we're seeing a great deal of complexity. And so we're seeing in some of these more recent phyla, you're seeing more development of the nervous system, and that gives them an advantage, and they pass that advantage on. And you also see in the cephalopods, they have a well-developed eye. And so again, a lot of the work that we do trying to understand how eyes work was done with cephalopods because their eyes are so well-developed, and they're very good. The cephalopod eye is much better than our eye at seeing underwater. Okay, so that's the Mollusca, just a brief introduction. It's a huge phylum. You could spend a whole class on just the mollusks, and some people do. We're going to be dissecting some of these, so we're going to look for some of these structures. If you've got any questions, let me know. That's all I've got, so I will see you in lab.